Dear students, after studying this module, you shall be able to know about the generation and regulation of enzyme reactions, learn about the nature of enzymes. You shall also be able to understand the food modification by enzymes, and you will be also knowing about the role of endogenous enzymes in food quality. So let us begin with this introduction of this topic, which is enzymes in food. What are enzymes? That is the first question that comes to mind. Enzymes are proteins with catalytic activity due to their power of specific activation and conversion of substrate into the products, as shown in this particular one reaction. Some of the enzymes are composed only of amino acids covalently linked via peptide bonds to give the proteins. Other enzymes contain additional components such as carbohydrate, phosphate and cofactor groups. Enzymes have all the chemical and physical characteristics of other proteins. Composition wise, enzymes are not different from all other proteins found in nature, and they comprise a small part of our daily protein intake in our foods. However, unlike other groups of proteins, they are highly specific catalysts for the thousands of chemical reactions required by living organisms. Enzymes are found in all living systems and make life possible, whether the organisms are adapted to growing near 0 degrees Celsius, at 37 degrees Celsius for humans, or near 100 degrees Celsius in microorganisms found in some hot springs. Enzymes accelerate reactions by a factor of 10 raised to power 3 to 10 raised to power 11 times that of non-catalyzed reaction. So that is a huge factor. In addition, they are highly selective for a limited number of substrates, since the substrate or substrates must bind stereospecifically and correctly into the active site before any catalysis occurs. Enzymes can also control the direction of reactions, leading to stereospecific products or product that can be very valuable byproducts for the food's nutrition and health or the essential compounds of life. Now, after having described to you some basics about the enzyme and its function, let us now study about the regulation of enzyme reactions. Enzyme activity can be controlled in a number of ways that are very important to food scientists. Velocity of an enzyme catalyzed reaction is usually directly proportional to the active enzyme concentration and dependent in a complex way on substrate, inhibitor and cofactor concentrations also, and also definitely on the temperature and pH involved in the reaction. As an example, it is well known that enzyme catalyzed reactions occur more slowly when a food is placed in a refrigerator, that is approximately the temperature of 4 degrees Celsius. But the reactions do not stop at 4 degrees Celsius or even at 0 degrees Celsius. Most enzyme catalyzed reactions decrease 1.4 to 2 times 10 degree decrease in temperature. Therefore, at 5 degrees, that is a 5 degrees Celsius, the velocities would be 0.5 to 0.25 times the velocity at 25 degrees Celsius. Changing the pH of the system by 1 or 2 pH units from the pH activity optimum can decrease enzymatic velocity to 0.5 or 0.1 respectively of that at the pH optimum. Decreasing the concentration of enzyme either by heating, breeding or genetic engineering to the concentration of enzyme to 0.1 that of normally present would decrease the enzyme activity to 0.1 that originally was present. Now we shall concentrate on the nature of enzymes. During the 1870 to 1890 period, that is the 2 decades, there was a major debate between Pasteur, who believed that enzymes functioned only when associated with living organisms, and Liebig, who believed that enzymes continued to function in the absence of cells too. The argument was settled in the year 1897 when Buckner separated enzymes including invertase from yeast cells and showed that they were still active. So, What does this mean and what are we going to study after this? We shall now concentrate our attention to the food modification by the enzymes. Enzymes have a very important impact on our quality of our foods. In fact, without enzymes, there would be no food at all. But then there would be no need for food also, since no organisms could live without enzymes because they are needed for the digestion purpose. They are the catalysts that make life possible as we know it. For any organism, life begins with the enzyme action in the gestation and fertilization processes. The growth and maturation of our foods depend on enzyme actions. While we have known for some time that environmental conditions during growing affect the composition, including enzymes of our plant foods, a recent review has detailed just how much effect moisture deficiency has on the expression of genes during growth and maturation of plants. Following maturation, the harvesting, storage and processing conditions can markedly affect the rate of food deterioration. Enzymes can also be added to foods during processing change their characteristics, and some of these changes will be discussed in this present module. Microbial enzymes left after destruction of the microorganisms continue to affect the quality of processed and reformulated foods. For example, starch-based sauces can undergo undesirable changes in consistency because of heat-stable microbial alpha amylases that survive a heat treatment sufficient to destroy the microorganisms. Because of their high specificity, enzymes are also the ideal catalyst for the biosynthesis of highly complex chemicals. So, what is the role of endogenous enzymes as discussed in the food quality? First role is the color. Color is probably the first attribute the consumer associate with the quality and acceptability of the foods. So let us discuss about the importance of color in foods. A steak must be red, not purple or brown. Redness is due to the oxymyoglobin, the main pigment in the meat. Deoxymyoglobin is responsible for the purple color of meat. Oxidation of iron 2 present in oxymyoglobin and deoxymyoglobin to iron 3 producing metmyoglobin is responsible for the brown color of meat. The enzyme catalyzed reactions in meat can compete for oxygen, can produce compounds that alter the oxidation reduction state and water content, and can thereby influence the color of meat. The quality of many fresh vegetables and fruits is judged on the basis of their greenness. On ripening, the green color of many of our fruits decreases and is replaced with red, orange, yellow and black colors. In green peas and English green peas, maturity leads to a decrease in chlorophyll level. All of these changes are a result of enzyme action. Three key enzymes responsible for chemical alterations of pigments in fruits and vegetables are lipooxygenase, chlorophyllase and polyphenol oxidase. Let us now discuss lipooxygenase. Lipooxygenase enzyme, which is a linoleate type of compound, falls in the oxidoreductase class of enzymes, has 6 important effects on foods, some desirable and the others undesirable. The 2 desirable functions are bleaching of wheat and soybean flours, formation of disulfide bonds in gluten during dough formation, which eliminates the need to add chemical oxidizers like potassium bromate. The 4 undesirable effects of lipooxygenase in food are destruction of chlorophyll and carotenes, development of oxidative off flavors and aromas often characterized as hay-like, oxidative damage to the compounds such as vitamins and proteins, and oxidation of essential fatty acids like linoleic acids and linolenic acids as well as arachidonic acids. All 6 of these reactions result from the direct action of lipooxygenase in oxidation of polyunsaturated fatty acids, that is free and lipid bound, to form free radical intermediates. Further, Non-enzymatic reactions lead to formation of aldehydes, including malonaldehyde and other components that contribute to off flavors and off aromas. The free radicals and hydroperoxide are responsible for loss of color, that is chlorophyll, the orange and the red colors of carotenoids, the disulfide bond formation in gluten of doughs, and damage to vitamins and proteins. Antioxidants such as vitamin E, propyl gallate, benzoylated hydroxytoluene, and nor-dihydroguaiacetic acid protect foods from damage from free radicals and hydroperoxides. The second in this category is chlorophyllase. Chlorophyllase is found in plants and chlorophyll containing microorganisms. It hydrolyzes the phytyl group from chlorophyll to give phytol and chlorophyllide. Although this reaction has been attributed to a loss of green color, there is no evidence to support this, as chlorophyllide is green also. The third in this category is the polyphenol oxidase. Polyphenol oxidase, that is 1,2-benzene-diol, falls also in the category of oxidoreductases, is frequently called tyrosinase, polyphenolase, phenolase, catechol oxidase, chrysolase or catecholase, depending on the substrate used in its assay, or found in the greatest concentration in the plant that serves as a source of the enzyme. Polyphenol oxidase is found in plants, animals and some microorganisms, especially in fungi. It catalyzes two quite different reactions with a large number of phenols. These reactions are: number 1, the 4-methyl-O-benzoquinone is unstable and undergoes further non-enzyme catalyzed oxidation by oxygen and polymerization to give melanins. The latter is responsible for the undesirable brown discoloration of bananas, apples, peaches, potatoes, mushrooms, shrimps, humans, and the undesirable brown and black colors of tea, coffee, raisins, prunes and human skin pigmentation also. The orthobenzoquinone reacts with the E-amino group of lysyl residues of proteins, leading to loss of nutritional quality and insolubilization of proteins. Changes in texture and taste also result from the browning reaction. It is estimated that up to 50% of some tropical fruits are lost due to enzymatic browning. This reaction is also responsible for the deterioration of color in juices and fresh vegetables such as lettuce, and in taste and nutritional quality too. Therefore, much effort has gone into the developing methods for controlling of polyphenol oxidase activity. So now we shall study about the texture. Texture is very important quality attribute in foods. In fruits and vegetables, texture is due primarily to the complex carbohydrates, that is pectic substances, cellulose, hemicelluloses, starch and lignin. There are one or more enzymes that act on each of the complex carbohydrates that are important in food texture. Proteases are important in the softening of animal tissues and high protein plant foods. Pectic enzymes. Let us discuss now about some pectic enzymes. There are three types of pectic enzymes that act on pectic substances, are well described in this module. Two pectic enzymes like pectin methylesterase and polygalacturonase are found in higher plants and microorganisms, and one type, the pectate lyases, is found in microorganisms, especially certain pathogenic microorganisms that affect the plants. Pectin methylesterase or the pectin pectyl hydrolase hydrolyzes the methyl ester bond in pectin to give pectic acid and methanol. Polygalacturonase is responsible for the hydrolysis of the alpha-1,4 glycosidic bond between the anhydrogalacturonic acid units. Both endo and exo polygalacturonases exist. The exotype hydrolyzes bonds at the ends of the polymer and the endotype acts in the interior. The pectinases splits the glycosidic bonds of both pectin and pectic acid not with water but by beta elimination. They are found in microorganisms but not in higher plants. A fourth type of pectin degrading enzyme, that is protopectinase, has been reported also in few microorganisms. Protopectinase hydrolyzes protopectin, producing thereby pectin. However, it is not clear yet whether the protopectinase activity in plants is due to the combined action of pectin methylesterase and polygalacturonase or to a true protopectinase. Let us now come to the discussion of cellulases. Cellulose is abundant in trees and cotton. Fruits and vegetables contain small amount of cellulose, which has a role in the structure of cells. Whether cellulases are important in the softening of green peas and English green pea pods is still a matter of controversy. Abundant information is available on the microbial cellulases because of their potential importance in converting insoluble cellulosic waste to glucose. Pentosanases. Hemicelluloses, which are polymers of xylose, that is xylans, arabinos, arabins, or xylose and arabinos, that is arabinoxylans, with small amounts of other pentoses or hexoses, are found in higher plants. Pentosanases in microorganisms and in some higher plants hydrolyze the xylans, arabins and arabinoxylans to smaller compounds. The microbial pentosanases are better characterized than those in higher plants. Several exo and endohydrolyzing pentosanases also exist in wheat at very low concentrations, but little is known about their properties. It is important that these pentosanases receive more attention from food scientists. Now we shall discuss students about the amylases. Amylases, these are the enzymes that hydrolyze starches. They are found not only in the animals but also in the higher plants and microorganisms. Therefore, it is not surprising that some starch degradation occurs during maturation, storage and processing of our foods. Since starch contributes in a major way to the viscosity and texture of foods, it hydrolyzes during storage and processing is a matter of importance. There are three major types of amylases: alpha amylases, beta amylases and glucoamylases. They act primarily on the both starch and glycogen. Let us now talk about alpha amylases. They are found in all organisms and they hydrolyze the interior alpha 1,4-glucosidic bonds of starch in both amylose and amylopectin, glycogen and cyclodextrins with retention of the alpha configuration of the anomeric carbon. Since the enzyme is endosplitting, its action has a major effect on the viscosity of starch-based foods such as puddings, cream sauces, etc. The salivary and pancreatic alpha amylases are very important in starch digestion in our foods. The beta amylases. They are found in higher plants. They hydrolyze alpha 1,4-glucosidic linkage of starch at the non-reducing end to give beta maltose. Since they are exosplitting enzymes, many bonds must be hydrolyzed before an appreciable effect on viscosity of starch is observed. Amylose can be hydrolyzed to 100% maltose by beta amylase, while beta amylase cannot continue beyond the first alpha 1,6-glycosidic bond encountered in amylopectin. Therefore, amylopectin is hydrolyzed only to a limited extent by beta amylase alone. Maltose syrups of about dP10 are very important in the food industry. Beta amylase along with alpha amylase is very important in the brewing industry since the maltose can be rapidly converted into glucose by yeast maltase. Beta amylase is a sulfhydryl enzyme and can be inhibited by a number of sulfhydryl groups and reagents unlike alpha amylase and glucoamylase. In malt, beta amylase is often covalently linked via disulfide bonds to other sulfhydryl groups. Therefore, malt should be treated with a sulfhydryl group such as cysteine to increase the activity in malt. Now students, we shall discuss about proteases. Texture of food products is changed by hydrolysis of proteins by endogenous and exogenous proteases. Gelatin will not gel when raw pineapple is added because the pineapple contains bromelain, which is a protease. Chymosin causes milk to gel as a result of its hydrolysis of a single peptide bond between phenylalanine 105 and methionine 106 in K-casein. This specific hydrolysis of K-casein destabilizes the casein micelle, causing it to aggregate to form a curd cottage cheese. Action of intentionally added microbial proteases during the aging of brick cheeses assist in development of flavors, that is flavors in cheddar cheese, that is blue cheese for example. Protease activity on the gluten proteins of wheat bread dough during rising is important not only in the mixing characteristics and also energy requirements, but also in the quality of the baked breads. The effect of proteases in the tenderization of meat is perhaps the best known and is economically most important example. After death, muscle becomes rigid due to rigor mortis caused by extensive interaction of myosin and actin. Through action of endogenous proteases, calcium 2 plus activated proteases and perhaps cathepsins on the myosin actin complex during storage, that is 7 to 21 days, the muscle becomes more tender and juicy. Exogenous enzymes such as papain and ficin are added to some less choice meats to tenderize them, primarily due to partial hydrolysis of elastin and collagen. Now we shall discuss about the flavor and aroma changes in food. Chemical compounds contributing to the flavor and aroma of foods are numerous, and the critical combinations of compounds are not easy to determine. It is equally difficult to identify enzymes instrumental in the biosynthesis of flavors, typical of food flavors, and in the development of undesirable flavors. Enzymes cause off flavors and off aromas in foods, particularly during storage. Improperly blanched foods such as green beans, English green peas, corn, broccoli and cauliflower develop very noticeable off flavors and off aromas during frozen storage. Naranjin is responsible for the bitter taste of grapefruit and grapefruit juice too. Naranjin can be destroyed by treating the juice with naranginase. Some research is underway to eliminate naranjin biosynthesis by recombinant DNA techniques too. Let us now focus our attention onto the nutritional quality. There is relatively little data available with respect to the effects of enzymes on nutritional quality of foods. Lipoxygenase or oxidation of linoleic, linolenic and arachidonic acids certainly decreases the amount of essential fatty acids in foods. The free radicals produced by lipoxygenase catalyzed oxidation of PUFA, that is polyunsaturated fatty acids, decreases the carotenoid, that is vitamin A precursors, tocopherols, vitamin E and vitamin C and folate content of many foods. The free radicals also are damaging to cysteine, tyrosine, tryptophan and histidine residues of proteins. Ascorbic acid is destroyed by ascorbic acid oxidase found in some vegetables such as squash. Thymidase destroys thiamine, an essential cofactor involved in amino acid metabolism. Riboflavin hydrolase found in some microorganisms can degrade riboflavin, and polyphenol oxidase, like discussed earlier, causes browning, decreases the available lysine content of proteins. Now we shall discuss with you students the enzymes used as processing aids and ingredients. Enzymes are ideal for producing key changes in the functional properties of foods, for removal of toxic constituents and for producing new ingredients. This is because they are highly specific, they act at low temperatures, that is 25 to 45 degrees Celsius range, and do not produce site reactions. So students, the first kind of enzymes in this category are the immobilized enzymes. Enzymes in solution are usually used only once. The repeated use of enzymes fixed to a carrier is more economical. The use of enzymes in a continuous process, for example, immobilized enzymes used in the form of a stationary phase which fills a reaction column where the reaction can be controlled simply by adjustment of the flow rate is the most advanced technique. Second category is the bound enzymes. An enzyme can be bound to a carrier by covalent chemical linkages or in many cases by physical forces such as adsorption, by charge attraction, hydrogen bond formation and or by hydrophobic interactions. The covalent attachment to a carrier, in this case an activated matrix, is usually achieved by methods employed in peptide and protein chemistry. Third is the enzyme entrapment. An enzyme can be entrapped or enclosed in the cavities of a polymer network by polymerization of a monomer such as acrylamide or NN-methyl bisacrylamide in the presence of enzyme and still remain accessible to substrate through a network of pores. Next we study the cross-linked enzymes. Derivatization of enzymes using a bifunctional reagent, for example glyceraldehyde, can result on cross-linking of enzyme and thus formation of large, still catalytically active insoluble complexes. Such enzyme preparations are relatively unstable for handling and therefore are used mostly for analytical work. Some important use of enzymes in foods. Let us now discuss this topic. There are some major successes in the use of food related enzymes. First one is the production of high fructose corn syrup and sweeteners. This involves a relatively heat stable alpha amylase, glucoamylase and glucoisomerase, that is glucose isomerase. You can see the two reactions here involving the enzymes. Starch is heated to 105 degrees Celsius. Bacillus licheniformis, an alpha amylase, is added and dextrins of DP10 to 12 are produced by the endosplitting enzyme. The soluble digest is passed through giant columns, that is 6 to 10 feet in diameter and 20 feet high, of immobilized glucoamylase where glucose is produced. The glucose containing stream is then run through giant columns of immobilized glucose isomerase where approximately equimolar concentrations of glucose and fructose are produced. Let us now discuss about specialty products and ingredients via enzymology. First, in this category we shall discuss about enzymatic removal of undesirable products. Raw food materials often contain toxic or anti-nutrient compounds that are sometimes removed by proper heat treatment, extraction or by enzymatic reactions. There are more than 12,000 plants in the world that have potential as food sources. Many are not used because of the undesirable properties, some of which could be overcome by the proper use of enzymes. Let us now discuss in this category particularly enzymes in milk and dairy products. Bovine milk contains many enzymes and other enzymes are added during processing. Of most importance economically is the use of chymosin, that is rennet, in production of several kinds of cheese. Beta-galactosidase is potentially of great importance in the commercial hydrolysis of lactose in milk and dairy products, so that these products can be consumed by individuals who are deficient in it. Beta-galactosidase is also used to convert whey lactose to glucose and galactose, sweeteners with greater commercial demand. Enzymes in baking. So let us discuss about this a little bit. Several enzymes are used in bread making. Additions of amylases and proteases has been common for years. Several enzymes preparations are available for the stated purpose of reducing the rate of staling in bread. So this table here shows you the enzymatic production of specific compounds or creation of desirable flavors in them. Let us now talk about, students, the enzymes in brewing. Several recent advances have been made with regard to the use of enzymes in the brewing industry. There is increasing interest in the possible use of the blended alpha amylase and protease preparations in replacement of malt in brewing. This is attributable to the expense and limited supplies of malt and the possibilities that better quality control might be achieved. The industry has also used amyloglucosidases recently to make light beer. Amyloglucosidases hydrolyze the alpha-1,6 glucosidic bonds of the amylopectin fraction, permitting the complete fermentation of the starch. Enzymes for the control of microorganisms. Enzymes have potential for destroying microorganisms by several means. These means range from hydrolysis of cell wall compounds such as beta-glucans, chitin and peptidoglycans to the production of hydrogen peroxide and oxygen, which oxidize the essential SH group of key sulfhydryl enzymes or polyunsaturated fatty acids in cell walls. These are interesting possibilities worthy of being tested at the commercial level. Now here table shows you the enzymes which are used for control of various microorganisms and their uses. What are the enzymes which are involved in waste management? Let us discuss about this. It is estimated that the annual production of major carbohydrate feedstock for the use as potential fuels or for manufacturing chemicals in the United States is about 1160 million tons versus 50 million tons of organic chemical feedstock. This number includes 160 million tons of municipal solid waste, 400 million tons of agricultural residue, 400 million tons of forest residue and 200 million tons of corn and grains. These residue pose major environmental problems since a large amount of agricultural residues are burnt where permitted. The forest residues result in major fires each year and the massive municipal landfills are often sources of groundwater pollution. Therefore, if some of these materials could be converted to other forms of fuel like ethanol or methanol or to fermentation feedstock like glucose to produce proteins, ethanol and CO2, disposal problems would be alleviated and non-renewable fuel sources or fuel supplies could be conserved. The US government is spending considerable money on research on how to efficiently tap these potential sources of fuel and chemicals. What are the compounds that need to be converted at this time? They are largely starch, cellulose, lignin, lipids, nucleic acids and proteins. What are enzymes needed for? And what are the enzymes? Primarily the amylases, cellulases, lignin peroxidases, lipases, nucleases and proteases. These are enzymes we know quite a lot about. So what is the problem? Where is the problem lying? The major problems are the insolubility of potential substrate just listed and the poor stability and catalytic inefficiency of the enzymes which are required. Some methods exist for increasing the solubility of the substrate and thereby the rate of hydrolysis. But all methods require considerable energy expenditures that makes the process uneconomical at the moment, when petroleum and coal are relatively inexpensive commodities. Do commercially feasible solutions exist for converting biomass to valuable chemicals? Nature does this efficiently, given the huge 1160 million tons to be converted annually. The natural conversion of these compounds occurs by action of usual classes of enzymes. But the process is very slow. Improvements in the rate are possible through redesign of the enzymes with greater efficiency and synergistic properties. Rapid economical biomass conversion is a difficult task, so the approach taken must be multifaceted. More efficient enzymes are needed, and this requires knowledge of why these enzymes are so inefficient in attacking insoluble substrate. Once this is better understood, better enzymes can be searched for or developed by recombinant DNA techniques. One needed property is greater stability. Especially at temperatures sufficiently high, microbial growth will not be a problem. The pentose containing polymers are especially difficult to hydrolyze and even more difficult to ferment to ethanol and carbon dioxide. Lignin severely limits hydrolysis and fermentation of cellulose because it is so inert. Enzymes that efficiently attack these substrates are the need of this hour. Solution of the biomass conversion problem is a must at this time. Conversion processes must be environmentally acceptable and all components, even minor ones, must be economically converted to usable or innocuous products to meet waste minimization goals pursued by the US Environmental Protection Agency. Now we have discussed enzyme in food to a great detail, students, and I would like to summarize this module for you. In this module, we have taught you that enzymes are proteins with catalytic activity due to their power of specific activation and conversion of substrate to products. Some of the enzymes are composed only of amino acids covalently linked via peptide bonds to give proteins. Enzymes have all the chemical and physical characteristics of other proteins. Composition wise, enzymes are not different from other proteins found in nature, and they comprise a small part of our daily protein intake in our foods. Some recent advances have been made with regard to the use of enzymes in brewing industry. There is an increasing interest in the possible use of blended alpha amylase and protease preparations as replacements for malt in the brewing. Use of beta glucanases may solve the high viscosity, low filtration rate problems caused by mannans from cell walls. The most exciting advance, however, is the use of acetolactate decarboxylase, now cloned into brewer's yeast, to shorten the fermentation time by avoiding diacetyl formation. Enzymes have the potential for destroying microorganisms by various means. This means range from hydrolysis of cell wall components such as beta-glucans, chitin, peptidoglycans to production of hydrogen peroxide and oxygen, which oxidize the essential SH group of the key sulfhydryl enzymes or polyunsaturated fatty acids in cell walls. It is estimated that the annual production of major carbohydrate feedstock for use as potential fuels or manufacturing chemicals in the United States of America is about 1160 million tons versus the 50 million tons of organic chemical feedstocks.